All right, so we're continuing talking about what these gene things are. And we've talked a lot about chromosomes and about fruit flies and the experiments done by Thomas Hunt Morgan and his students. Utter trivia that I'd never test you on, but Morgan's grandfather was actually a Civil War cavalry commander on the Confederate side, and did a lot of very daring raids and things like that. Uh, Thomas Hunt Morgan actually encountered a professor in his school years uh, who'd served on the Union side and had actually had to fight his grandfather, and that gave him kind of bad moments, but the professor did end up passing him. Okay, Cooper, thanks for the singing, but we'll save that. Save that till after we're done. You can rip off an aria or something. Okay, so we now shift from fruit flies to bacteria. To London in the year 1928. There's a British bacteriologist, guy who studies bacteria, named Frederick Griffith, working for the British government in public health, studying a bacterium called Streptococcus pneumoniae, which causes bacterial pneumonia. That's a lung infection. Since it is coccus, it looks like tiny dots. Strepto means when it grows, it tends to form little chains of cells. And pneumoniae means it causes lung infections. Now, you might remember way back when I was talking about bacteria, I happened to mention casually that some bacterial cells were surrounded outside the cell wall by what we call a capsule, basically a layer of slime surrounding the actual cell. And this is what it looks like on a stain slide. The little dark dots are the actual bacteria. Surrounding those dots, you have this light colored halo that doesn't take up stain. That's the capsule. The reason why this matters from a medical point of view is that bacteria with the capsule have some protection against your immune system, specifically the white blood cells that fight bacterial infections by engulfing and digesting the bacteria. Bacteria with the capsule are protected against that, and so they cause severe disease because they can dodge the body's main defense. Bacteria that don't have that capsule are less likely to cause serious disease because your body's immune system can handle them much better. That's not necessarily true for all bacteria, but it is for Streptococcus pneumoniae. Brendan, your mic's on. Okay. I think in an upcoming lab, you're going to get to work with a substance called agar. Agar is harvested from seaweeds, of all things. Agar, when you boil it with water and then let it cool, it forms a gel. Looks a little bit like Jello, about the same consistency too. Incidentally, agar is edible. There's some Japanese desserts. I think it's called kantan. I might have that wrong, but there's some Japanese desserts that are made with fruit flavored agar. But in the West, it's usually used to grow bacteria. You'll pour some liquid agar into a little dish, let it harden, and then you can spread bacteria over the surface of the agar. The reason you'd want to do that is that each bacterium on an agar surface will divide and divide again and again and again, but the bacteria can't eat the agar itself, and most of the time they can't move over the surface. Which means that if you spread bacteria over the surface of a dish of solid agar, what you get is little clumps of bacteria. Each clump is millions of bacterial cells growing in the same place, dividing and making more, but unable to spread out and move around. And each one of those little clumps is called a bacterial colony. I used to do this sort of thing all the time, spread samples on plates and then look at them within about 24 hours to see if colonies had grown out. I used to do that all the time when I worked at the Berkeley Free Clinic in grad school. Your local doctor does this all the time with things like throat swabs, sometimes urine samples if you have a urinary tract infection, things like that. Anyway, you can tell the difference between Streptococcus pneumoniae that doesn't have the capsule and Streptococcus pneumoniae that does, because the same bacteria can vary. It can have or not have the capsule. Strep pneumoniae with no capsule make these little rough-looking colonies that we call type R, stands for rough. Streptococcus pneumoniae make much bigger colonies, and because the bacteria are covered in this slime, the colonies are not only larger, but they look glossier. They look like drops of a very smooth and glossy slime on the surface of an agar plate, and we call that type S for smooth. Okay, so what? Griffith injected S-type bacteria into mice, and the mice died of the infection within 24 to 36 hours. More to the point, you could take blood samples from the dead mouse and spread them on an agar plate, and you got S-type bacteria back. No real surprises there. You inject live S bacteria, they kill the mice, and you find the mouse is full of S bacteria, right? No surprise at all. The R strain of bacteria, you inject those into the mouse, and the mouse is usually okay. The mouse might retain a few R-type bacteria inside it, but most of those get mopped up by a healthy mouse's immune system and don't cause the mouse any permanent harm. Okay, so S bacteria are lethal, R bacteria are mostly harmless. Okay, if you take S bacteria that you've killed by heating them and inject them into the mouse, you get nothing, right? You injected dead bacteria, the mouse's immune system basically cleans those up. There's no infection, the mouse is healthy, and the mouse doesn't have any living bacteria in it. What Griffith found was that if you mixed live R and dead S, that did kill the mice. Think about this, live R bacteria don't have the capsule and they don't kill the mouse. The mouse's immune system cleans those up. Dead S bacteria don't kill the mouse because they're dead. But mixing live R and dead S does kill the mouse, and the mice were found to have live S in their bodies. So either the dead S bacteria came back to life like zombies, which they didn't, or your other option is that some substance had gone from the dead bacteria to the living ones that transformed them from R to S. The dead bacteria might have been dead, but they must have had something in them that somehow got picked up by the living R type and it turned them into the S type. There's something that can actually change a bacterium's phenotype. What he called this was the transforming principle, which raises the question, what exactly is the transforming principle? That question was picked up by the top American specialist in Streptococcus pneumoniae, a man named Oswald Avery. who started repeating Griffith's experiments, but instead of turning R into S bacteria with whole dead S bacteria, they started killing S bacteria, breaking them up, separating them into different substances, and testing which one would cause the transformation of R-type bacteria. And it took them 15 years of work, but they finally isolated what the transforming principle was, what chemical inside bacteria actually did this. It turned out to be something that scientists already knew about. It had been discovered back in 1869. It was known to be found in the nuclei of eukaryotic cells. Prokaryotes have it too. And when you isolate it, it's a very weak acid. So because it was an acid and found in the nucleus of eukaryotic cells, it was called, dun dun dun, nucleic acid. Ooh, ah. Bear in mind that prokaryotes have this too, even though they don't have nuclei by definition. Their cells do contain nucleic acid. And it turns out that nucleic acid is one of two major substances that chromosomes are physically made of. The other one is a type of substance called protein. We'll talk more about what proteins are later. I think you've already worked with them in that food chemistry lab that you did. But Avery was able to show that protein was not the transforming principle. What actually does the trick of transforming bacteria was this nucleic acid. This test tube, by the way, shows nucleic acid that's been isolated from cells, and it's that pinkish stuff with little bubbles in it right in the center of that test tube. If you look carefully, the fluid in the upper and lower parts of the test tube is mostly colorless, but there's pink stuff in the middle. That's what nucleic acid looks like when you extract it from eukaryotic cells. Okay, what's nucleic acid? I've got to backtrack a little bit and talk some chemistry. Some of you may know this already. Again, I'm not trying to talk down to anybody, but I want to make sure that I don't leave any gaps. We know that matter is made up of tiny little particles called atoms. In nature, there are 88 naturally occurring types of atom. In other words, there are 88 substances that we call chemical elements. There's also over 30 elements that have been made only in laboratories. We're not going to go into them. They're all radioactive, and aside from being some of them quite dangerous, they don't play much a role in biology. So atoms can stick to each other. They can form atomic bonds. And when they do that, we call the clusters of atoms that develop molecules. And the material that's made of molecules is a chemical compound. Very simple example, the salt in your salt shaker is a chemical compound because it's made of atoms of sodium stuck to atoms of chlorine. And so the compound is sodium chloride, otherwise known as table salt. Right. That's a diagram of a compound that happens to be called styrene. It's made only of atoms of the element carbon and the element hydrogen. The carbon atoms make a six-sided ring. That's a very common feature of compounds that are made of carbon. They make a lot of rings. And then sticking up from that top carbon at about the 10 o'clock position, you have two more carbons that are stuck to that ring carbon, and they've all got hydrogen stuck on them. And when carbon atoms link up with each other and form bonds, They can be single bonds or double bonds, or in some cases, triple bonds. That's why some of these carbons are connected with single lines and some are connected with double lines. For what we need to do here, we don't need to worry about what those bonds are. We've got a perfectly good chemistry department to teach that should you need to know. For what we need to do here, you can pretend that a carbon atom has 4 little hooks sticking out of it, and each one of those hooks needs to hook up with the hooks on another atom. Hydrogen atoms, you can pretend, are little balls that only have 1 hook, and oxygens have 2 hooks, nitrogens have 3, and carbons have 4. That's not accurate, but for what we need to do in here, it will do. So I can take a bunch of styrene molecules and I can line them up like this and then cause new chemical bonds to form. And the result looks like that, where I've taken that little 2 carbon group that's sticking off the ring and stuck it to the 2 carbon group on the next molecule. And then that one sticks to the one after that. And I make this long chain of styrene molecules, each of which is stuck to the next. When I do that, I have a compound called polystyrene. Now, solid polystyrene is used to make everything from like the little cups that you get yogurt in. If you buy those single serving yogurt cups at the grocery store, those are made of polystyrene. If any of you still remember compact discs, the jewel cases, the plastic cases that the disc and the little booklet come in, that's polystyrene. And then if you make a batch of polystyrene and blow air into it, so you make a bunch of tiny bubbles, you get a very light substance called styrofoam. So everything from insulated coffee cups to packing peanuts to cheap coolers for taken out on the river are made of styrofoam, and that's just polystyrene with a lot of air bubbles added. Anytime we have a large molecule that's made up of repeated subunits that are strung together in a long chain, we call that a polymer. So polystyrene is a polymer. PVC, that stuff that a lot of pipes get made of, that's a polymer. It's called stands for polyvinyl chloride. All of your plastics are polymers. Lots of biological molecules are polymers as well. They're big molecules made up of a long chain of identical or very similar smaller molecules. The subunit molecules, by the way, are called monomers. You don't have to know this, but mero in Greek means a division. So a monomer is made up of 1 division, right? That's a monomer. And when you take a bunch of them and you string them together, you get a polymer, many divisions. And you can think of polymers like a train. I assume you've all seen trains, like especially if you are, driving across downtown Conway and in a real hurry to get somewhere. You've probably seen the trains that come through town. And the next time you get stuck while a train is driving right across Oak Street, consider it. It may be a really long train, but there's only a limited number of cars that make it up. You'll see box cars, you'll see flat cars, you'll see those piggyback freighters. Those are the ones that have those big shipping containers that you can take off the train and drop right onto an 18-wheeler. You might see tank cars for liquids. If it's a passenger train, I don't think we get any of those coming through Conway, but you might see Amtrak Dreamliners. You'll see a limited number of car types, but you can put them together in any combination that's convenient. You could have a train that's all one type of car. You could have a train that's four different types of car. You could put all the box cars together. You could alternate box tank, boxcar, tank car, boxcar, tank car. Whatever it is that you want it. You can put a limited number of types of car together into a train that can carry any combination of loads that you want to, in any combination that is convenient for your purposes. Polymers work like that. You might have only one type of subunit, or you might have a limited number of types, but you can string them together in any combination. And polymers are very important in organisms. Proteins are polymers of smaller molecules called amino acids. We'll get to those later. Cellulose is a polymer of simple sugars. Each one of these hexagons with an O in it is a ring of carbon and hydrogen atoms plus an oxygen. And I should mention that carbon is so abundant in the chemistry of life that we often don't even bother to write in all those letters C that stand for a carbon. Basically, anywhere you've got an angle in this diagram, there is a carbon atom right there. It's so abundant that we often leave them out completely. So each of these hexagons is a ring of carbons and hydrogens with a couple of oxygens thrown in, and they're strung together from one end to the other. Real cellulose might contain hundreds, if not thousands, of these carbon and oxygen rings strung together from one end to the other. Makes it a very, well, when you eat cellulose derived from plant cell walls, when it's in your diet, you call it dietary fiber. And the reason why that stuff is fibrous is that it's made of these long chains of sugar molecules. It makes it very tough and, well, fibrous. The chitin in fungi is a slightly different polymer of these ring-shaped molecules. And I should add that these rings of carbon with oxygen stuck into them are simple sugars. The plant makes sugars by photosynthesis. One thing it can do with them is stick them together to make cellulose, which is very tough and very resistant. Most organisms can't break it down. Termites, however, can, but that's another story. The reason this matters is nucleic acid also turns out to be a polymer, and its structure had been worked out in 1919 by J. Rando scientist. His name was Phoebus Levine. I'm not going to test you on his name. And he described the structure of the nucleic acid monomer. So a nucleic acid is a long, long chain of molecules that look like this. Molecules that are what we call nucleotides. So this is what a nucleotide looks like. And nucleotides themselves have parts. Just like every train car has wheels and axles and a base, and then whatever it's carrying up above, the tank or the refrigerated compartment or whatever it might be, just as every train car has parts, every nucleotide has parts. Over in light yellow, over there on the left, there is a phosphorus atom. Remember, this is the P in schnapps. This is why you need that phosphorus. This is why you need P. And that phosphorus group is stuck to a bunch of oxygens and a couple of hydrogens for flavor. When you get a phosphorus stuck to 4 oxygens like this, we call it a phosphate group. The phosphate group is stuck to a sugar, which is outlined here in green. That sugar is a ring of carbons. And again, we don't write all of the carbons, but there's a carbon atom everywhere there is an angle in this diagram. And there's some hydrogens and oxygens stuck in there. That is a simple sugar. If you ate it, it wouldn't necessarily taste sweet. Not all sugar, the sugar that you put on your cornflakes in the morning is of a type called sucrose. The simple sugar that gets used in a lot of sodas these days is fructose. The sugar that diabetics monitor is glucose. They're all a little different. There's lots of other sugars. This one happens to be called deoxyribose. Stuck to that sugar, you have a ring of carbon and nitrogen atoms. In this case, it's a double ring. And this double ring of carbons and nitrogens is called the base. And we've highlighted it here in blue. So every nucleotide consists of a phosphate, a sugar, and a base. And nucleic acids are made of lots of nucleotides strung together in a long line. Incidentally, this sugar right here, if it had another oxygen sticking onto it, right where the red arrow is pointing, the sugar would be called ribose. I can't remember. I think ribose has to do with the name of the plant that it was originally isolated in. It's not really critical what it means. But if there was an oxygen right there where that red arrow is pointing, we'd call it ribose. Since there is an oxygen that is missing right there, we call that sugar deoxyribose. D means we took it away, right? As in we take something away. If you're deprived or demoted or deloused or something like that, that means taking it away. Oxy for oxygen, deoxyribose. And if you take a bunch of nucleotides that use deoxyribose and you string them together, your polymer is called deoxyribonucleic acid. Or if you don't feel like saying deoxyribonucleic acid, you can just call it DNA. So there, that's what DNA stands for. This is stuff I know you've heard of. It's in the news, it's in horror movies. You hear a lot about DNA testing and things like that. We'll get to that in a bit, but I'm going to go through exactly what this DNA stuff is, because figuring out what it really is critical to figuring out how it works, and that's what will actually put all of the jigsaw pieces together in the end. This is what's going to link Mendel breeding his peas and Van Beneden staring at worm chromosomes. This is where it all comes together. It turns out that we know DNA is the transforming principle. We know it's a polymer of nucleotides. We also know that not all of the nucleotides are the same. In fact, there's not one different nucleotide in DNA. There are 4. They all have the same phosphate and sugar. Where they differ is in what base they've got. And in fact, there are 4 bases. 2 of them that are rings of mostly carbon and nitrogen atoms, adenine at the upper left and guanine at the lower left. And 2 of them are single rings of carbons and nitrogens and an oxygen or 2 stuck on. We call these thymine and cytosine. And we just abbreviate these as A, T, G, and C. And it turns out that the bases are key to understanding how DNA works. In fact, they are so important that biologists have a song that they sing about them. And it goes, I'm all about the base, about the base. No sugar. I'm all about the base, about the base. No phosphate. Thank you. Thank you very much. I'm sure you're applauding wildly. So, yes, biochemists, when they study DNA, are definitely all about the base. Okay, why? 1940s, there's an Austrian biochemist by the name of Erwin Chargaff who discovered some facts about DNA. He took DNA from different species and broke it down and discovered that not all DNA was the same. For one thing, different species have different percentage composition of nucleotides. DNA is not 25% each, A, T, G, and C. Human DNA contains roughly 29% DNA. Octopus DNA is about 33%. Corn is about 27%, and so on. The actual numbers aren't really important. They're just numbers, but they do show that DNA is different in different species, which, well, you might expect. We know that DNA has something to do with heredity because it's what's in the chromosomes that has something to do with making the phenotype. So the reason why you don't look like an octopus must be because you and an octopus have different DNA. So no surprises there. But Chargaff found that the percentage of adenine was always equal to the percentage of thymine. The percentage of guanine was always equal to the percentage of cytosine. So if your DNA has about 29% A, it also has 29% T. Okay, great. 29 and 29 is 58. So 58% of your DNA is A and T. That leaves 42% of your DNA is G plus C, right? Because all the percentages have to add up to 100, right? 58% of your DNA is A plus T. 42% of your DNA is G plus C. And the percentage of G must be equal to the percentage of C. So your DNA is 21% G and 21% C and 29% A and 29% T. So if you know how much you have of one nucleotide, you can always calculate the percentage of the other three. And we still use these. They're called Chargaff's rules. Step forward to the early 1950s. Here's a British scientist on the left by the name of Francis Crick and a visiting American by the name of James Watson, they're both interested in DNA and they're working at Cambridge University in England in the lab of a senior scientist named Maurice Wilkins. They know DNA is important. And one of their colleagues I finally get to talk about a female scientist now. I know this has seemed like the March of the Dead white guys, but at least we've got some women coming up that we can talk about. And we'll get to some non-white people as well, I promise. This is Roz Franklin, and she was in their lab, and she was one of the world's leading experts in a technique called X-ray crystallography. What the hell is X-ray crystallography? How to do this had been discovered a couple of decades before. In fact, the discoverers won one of the early Nobel Prizes in, I think, chemistry. And the way it works is you take a substance that you want to study. It could be as simple as common salt, or it could be as complex as DNA, and you make a crystal out of it. which basically means that you line up all of those molecules in a very regular pattern, and then you hit that crystal with a focused beam of X-rays. What happens is my grandmother used to have hanging in her windows these little glass ornaments called sun catchers, right? They look like giant diamonds. They've got this very regular geometric shape. And every time a sunbeam hits one, the sunbeam passes through the sun catcher and gets broken up and makes this pretty little pattern of rainbow spots on the wall. She had chandeliers that would do the same thing. Those little pieces of glass on a chandelier, uh, when light shines through one of them, it gets broken up into a bunch of pretty little spots, right? I'm sure some of you have grandmothers that have the same thing. You could take a crystal, heck, take a diamond if you have a diamond ring, and you can see this, that crystals tend to break up light and break it up into these patterns of spots. Well, the same thing happens when you take a crystal of something and you hit it with X-rays. The X-ray beam breaks up, and it breaks up into a bunch of beams that hit, in this case, a detector. You can't see X-rays, but you can detect them with a big sheet of photographic film. And you get a picture that looks like this. The hard part is that you can measure the positions and the distances of those dots on the film, and you can use that to work out the position of the atoms in the crystal. This is really hard to do. It takes pretty gnarly math, and Ross Franklin was one of the best in the world at this technique. And it was the first way we were able to start working out the shape of molecules. Much too small to see molecules, even with the most powerful light microscopes. With all microscopes, except for a very few, you can't see individual atoms or molecules, but you can use this to work out the arrangement of atoms in a molecule and work out the molecule's shape. And I will be coming back to this again and again. This matters because if you understand the shape of something, you understand what that something can do. Just like the shape of a screwdriver or a wrench or a hammer or any other tool is a clue to understanding what that tool can do if you use it. The same thing is true for molecules. Working out the shape is critical for working out the function. There's Franklin on the left. On the right is an X-ray crystallography picture that she'd taken of using a crystal of DNA, and it looks like a bunch of blobs. She was able to work out that the DNA molecule must be shaped like a coil of some kind, like a very long spring. She was actually able to work out about how wide the spring was and how tightly coiled it was, what the distance was between coils. So she's able to work out the shape of the spring. And the word that we use, by the way, for a coil shape is a helix. So a corkscrew is a helix, a bed spring is a helix, a snail shell is a helix. Um, one of those door stops might be a helix. Any kind of coil shape is a helix. And Franklin was able to work out that DNA, whatever it was shaped like a helix. And she was able to work out how narrow it was and how tightly spaced it was, and a few other clues to what DNA's shape must be. Watson and Crick got hold of Ross Franklin's data, and be it said they did this without her knowledge or permission. They knew the DNA molecule was a helix, and they knew how wide and how tight it was. They knew the phosphate groups had to be on the outside. They knew how big nucleotides were. They knew what shapes they had, and they knew about Chargaff's rules. So they tried putting together models that would fit all of these criteria. We know the parts, we know what the complete product is supposed to look like. How do we fit the parts together to make the complete product? Maybe you've assembled things like that. You know, okay, I just bought a kit. I have the parts. I can see on the box what the kit is supposed to look like. How do I make the parts fit together to make what looks like the box? It's not a great way to put a bicycle together, but it worked for Watson and Crick. And they published a paper that described their model for the shape of DNA. And it turns out DNA is not a single polymer, it's two polymers of nucleotides wrapped around each other, forming a structure called a double helix. There they are pointing at a model that they've made of the double helix. And again, if you're wondering why the hell this matters, that double helix shape is critical for understanding how DNA does what it does. This is the key to solving the riddle of heredity that we'll be going through this week and the week after break. These might be a little bit easier to see than that picture. Those are computer models of DNA. They're the same molecule, one with the atoms represented as little sticks and one with the atoms represented as little balls. It's the same thing. What I want you to see is that the sugars and the phosphates are colored white and pink here. The bases are colored red and yellow. And you can see there's two chains of sugars and phosphates that wrap around each other. And the bases are in between those sugars and the phosphates, and they lock the two chains together. If we could untwist a bit of DNA, it would look like a rope ladder. The sugars and the phosphates are what stick to each other, and they form the sides of the ladder. They alternate. Each phosphate bonds to a sugar, which bonds to a phosphate, which bonds to a sugar, and so on. That's the ladder sides. The rungs of the ladder are made up of the bases, and the bases bond to each other to form the rungs. So take a rope ladder and then twist it in a spiral, and you'd have a decent model of DNA, a double helix. Somebody text me how I'm doing on time. I just realized I can't see my phone, and I don't want to go long. Am I starting to run out of time?
It's 10:48.
10:48. Okay. So there's the untwisted DNA molecule that looks like a ladder. And there's the twisted DNA molecule, and that's the double helix shape. Here, the sugars and phosphates have been replaced by these two things that look like ribbons to make it easier for you to see the bases on the inside. In fact, this is jumping way ahead. This was done about 20 years ago. There's a technique called scanning tunneling microscopy. Never mind how it works. It wasn't known back in Watson and Crick's day, but it's a technique that actually lets you take pictures of actual molecules. You can see molecules and even atoms with scanning tunneling microscopy. And there's a diagram on the left. Again, the sugars and phosphates have been mostly replaced with those things that look like ribbons to bring them out and make it look more clear. And you have the bases in the middle between those two strands. On the right, that's an actual picture of a DNA molecule taken using scanning tunneling microscopy. And it's not super clear, but I think you can see that it's got the same kind of twisted or spiral shape, and you can even see bases in between. So Watson and Crick basically got this right. Watson, Crick, and their lab supervisor won the Nobel Prize for Medicine. Rosalind Franklin probably should have shared the prize, except she died in 1958. People who work with high energy X-rays tended to die kind of young because they also cause cancer. The Nobel Prize is not given posthumously. It's not given to people who have died. So technically, she could not have shared the prize, but she doesn't always get credit when it was her data that was absolutely critical in working out the double helix shape. And that's a pretty good place to stop, although I'll have a little bit more to say about her when we convene on Friday. So we